Unit 2, page 3, Diffusion and Osmosis, Cellular Transport. Do now. Even though you weren't in the kitchen, you know that your friend is baking cookies. Explain how this is possible. Take a moment, write your answer down on the lines on your notes handout. So hopefully you said you smell the cookies. Duh. But why do you smell the cookies? Well, when we heat things up, a lot of times there's an odor that is created, and that odor can travel through the air to other places, maybe in your house or even outside. We're going to talk about diffusion today, as well as osmosis and active transport. So let's define diffusion. It's defined as the spontaneous movement of a substance down its concentration gradient from a more concentrated to a less concentrated area. This is when particles are spreading out and they equalize concentration throughout the available space. If you look at the picture on the right, you can see the food coloring is being added to water, and it's most concentrated immediately at the very top right now, and then it spreads throughout until it evenly distributes itself in the glass. This is diffusion. Cellular transport is defined as the movement of molecules in and out of a cell. So diffusion actually occurs inside and outside of our cells, as well as in regular settings like the food coloring or the cookie example. Today we're going to talk more about the cellular definition of diffusion, and there are two major types. There's passive transport, which does not require energy, and it moves down a concentration gradient. So it's when molecules are spreading out. Also, the other one is called active transport. Notice active and passive have opposite meanings. So active does require energy, and it moves up a concentration gradient. So question. From the last lecture, you should know the answer to this. Which organelle or cellular structure is involved in transporting materials in and out of the cell? Hopefully you knew cell membrane is the organelle that does that. Passive transport is the movement of molecules across a membrane down the concentration gradient. So that's when molecules go from high to low concentration without using any energy. Notice that in the picture, molecules are moving back and forth, but on average, they start out on the left, more of them are on the left, and they're kind of moving so it's an even amount of molecules on the left and right, and those purple circles are representing the molecules. So our concentration gradient, again, goes from high to low. High just means there's a lot of them, low, there's very little. Diffusion is the movement of molecules across a membrane without using energy. Molecules move down a concentration gradient until equal on both sides. Notice the same picture. So you see less movement occurring as they start to balance out on both sides. Again, this structure in the middle is representing the cell membrane. Osmosis is a specific form of diffusion, and it's actually when it occurs with water molecules. We'll be observing these in several labs in this class, but you can see pictures of it below. It equalizes the concentration on both sides of the membrane. So you can see in the picture on the left, there's a sugar solution. And you can see there's a membrane placed in the middle of the beaker. Most of the sugar solution is located on the left side of that membrane, and there's very little on the right. So over time, what's going to happen is, because there's more water on the right-hand side, you see water moving to the left side, right? Because there's more water in comparison to sugar. They're moving to the left. It's a little bit confusing because if you're too focused on the sugar molecules, those circles, you might not remember that water actually is also moving. Facilitated diffusion is diffusion of molecules across a membrane with the help of a protein. So if you look in the picture, you can see these big purple oval shapes, and those are proteins on the surface of the membrane. So we're zoomed in very close on a cell. This is the membrane here with the squiggly lines and the circles, and we're looking at it kind of cut through. So this is the inside of the cell down here. The blue part is outside of the cell. These proteins allow the movement of these molecules. So you can see some molecules have to go through this protein channel, like the orange triangle and the blue circle. But other molecules sometimes don't need a protein molecule, like the purple circles, and they can just go through. If they just go through, that's diffusion. If it requires a protein channel, that's facilitated diffusion. It needs a little bit of facilitation, a little help. So there are many channels for specific molecules. So only the triangle can go through, and only active transport is basically the opposite of passive transport or diffusion. It's the movement of molecules across the membrane by using energy. So that's where it gets its name, active transport. The movement of molecules are always going against the gradient, so they're going from a place where there's not very many, it's low concentration, to a place where there's lots of them packed together, high concentration. So you can see in this picture, there's an image and it says ATP with kind of an explosion looking thing around it. ATP is energy. So you might want to label that in your notes as a form of energy that the cell uses. Notice that energy is being used to push more of these orange squares inside of the cell. Notice there's already a lot of orange squares inside the cell, but even more are being pushed in. That's active transport. Those molecules don't want to be all squished together, but the cell, for whatever reason, really wants those molecules. So maybe it's a nutrient that the cell needs to survive. Next, endo and exocytosis. These are two processes where materials are either going in or out of the cell. So endocytosis, the prefix endo means inside or within, means to take in materials into the cell. And it basically folds the cell membrane inward, so you can see this little green piece of food. And the cell is down here, it's actually being engulfed, so it's taking the membrane and forming this little horseshoe shape. And eventually it pinches off so that the food comes into the cell and gets digested as nutrients for the cell. Exocytosis is the opposite. So it would be the excreting materials from the cell, releasing them at the cell membrane. So imagine this going the other direction, taking something and basically kind of spitting it out on the other side. So this is another job of the cell membrane, as well as diffusion and active transport. So summary, cell transport scenarios. Scenario 1, you have a picture of a cell and there's different molecules located inside and outside the cell. The question is, what would most likely happen over time? So in order to answer this question, we've got to identify all the molecules first. So how many different molecules do we have and what are their names? We've got carbon dioxide and oxygen. Carbon dioxide is represented by CO2 here, and oxygen is represented by O2 here. Next, we have to count the amount of each type of molecule. So if we look at CO2, how many carbon dioxide molecules do we have? Hopefully you located 5 of them. How about oxygen? 7. So we made a little chart, and you can see we've got our 2 types of molecules. We've got the amount inside and outside of the cell. So how do we decide what's inside and what's outside? If you look at our picture, inside is anything within this circle, and outside is anything beyond it. So here are our numbers. We've got 4 carbon dioxide inside, 1 outside, 2 oxygen inside, 4 outside. So how do we balance them? For carbon dioxide, there's more inside than outside. So the movement has to go out. For oxygen, there's more outside than inside, so those molecules will move in. So you could write something like, some carbon dioxide molecules move outside of the cell, and some oxygen molecules move into the cell, until all molecules have equal amounts both inside and outside the cell. Remember, the objective is always molecules inside and outside of each type. So you want the same amount of oxygen inside as well as outside, and the same amount of carbon dioxide inside and outside the cell. Scenario 2 shows two pictures, two different types of cells with dark dots indicating small molecules. The molecules are moving out of the cells as indicated by the arrows. Notice they both have arrows pointed out, but the dots are not the same in both pictures. So we're going to describe the difference in concentration inside and out of cell B and A. Start with cell A. Concentration inside, is it high or low? Hopefully you said low. If we count up the number of dots, there's only 4 of them. Outside is high because there's 10. So 10 is greater than 4, so we'll identify as the outside is high and the inside is low. What direction should the molecules be moving? Into the cell. Cell B. Concentration inside, is it high or low? It's high. Notice that there are 9 molecules inside, and what about 5 molecules outside, so 5 is low. So molecules should be moving out of the cell. Next question says, which situation would require ATP or energy? And the hint here is that which orange arrow went the opposite direction than the picture shows in the black. So if we look, here our orange arrow is going in, but the black arrow is going out. In cell B, they're both going out. So cell A is the one that requires energy. In order to push those molecules out, it requires extra energy because they ordinarily want to move inward.